very much. So I will walk you through the efficacy data of the trial. Next slide, please. So here is a summary of some assessments that we did during the trial. Efficacy was assessed at screening prior to the treatment start and during the treatment period. 3 days before every infusion visits, the patient fill out the patient home diary, where they every half an hour indicated whether they were on without dyskinesia, on with non-troublesome dyskinesia, on with troublesome dyskinesia, off or asleep. Every 3rd month, we assess the patients with the UPDRS, Unified Parkinson Disease Rating Scale, which is the golden standard in our field. The part 1, 2, and 4 were assessed in on, and they relate to the mental state, activities of daily living, and complications of therapy. The motor assessment was done in the part 3 was done in off. We also assessed the patients in off in the time up and go walking test. And the patient filled out the quality of life questionnaire called PDQ-39, and we assessed them with the clinical global impression scale. Next slide, please. There were also other exploratory efficacy assessments. That PET imaging to assess the dopamine system was performed at several time points during the study. We analyzed the blood serum and cerebrospinal fluid for CDNF levels and alpha-synuclein levels in CSF. And there is also an initiative to find the new biomarkers in these biofluids that may be affected by CDNF. We used an accelerometer, Parkinson's kinetograph, PKG, which is a wrist-worn moment measuring device that was worn 6 days prior to each infusion visit. Next slide, please. So here are the data from the motor rating using the UPDRS part 3 in off. In the dotted red line, you can see the expected annual decline and the worsening of the Parkinsonism in this population of Parkinson patients that has been found in the literature and in previous work. In gray, you see the placebo, in green, the mid-dose CDNF effect, and in blue, the high-dose CDNF. As you can see, there was a small placebo effect in the beginning of the study, but then went back to some extent. In green, you can see that the mid-dose CDNF has a lower rating than the expected annual decline. And when we combine the mid-dose and the high-dose CDNF ratings, we actually have fulfilled the definition of a minimal clinically important difference in this motor rating of UPDRS. Next slide, please. So there was a lot of variation between the patients, but there were a few responders, and here is one case. So this is a patient who came into the study with at Hönen-Jahr stage 2 with a disease duration of 10 years. He or she had an UPDRS motor rating in off at 25 with a very good levodopa response. So after levodopa treatment in on, the UPDRS part 3 was 11. When we blinded the data, we could see that this patient had received a placebo with an initial small response but then got higher UPDRS scores again. But we could see with the mid-dose CDNF that there was a clear improvement on UPDRS motor rating in off. That actually led the clinicians for this particular patient to reduce the anti-Parkinsonian dopaminergic therapy. Next slide, please. This is an example of the initial analysis of the accelerometer data with the PKG. And it is the bradykinesia score, the slowness of movement score, that is depicted here. And you can see in red the expected increase in bradykinesia for a patient on vehicle. Whereas for the CDNF, and especially at the mid dose, we don't see this worsening of scoring with the accelerometer over the time of the main study. And there is actually a significant difference between the mid-dose CDNF and vehicle with this accelerometer measure. And more data will soon be released based on these assessment that we did. Next slide, please. Then another exploratory measure was the DAT PET, and here are examples from 2 patients at baseline and after 6 months of within the study. At baseline, you can see that there is a very, very strong reduced level of the DAT signal in putamen with some signal still in nucleus caudatus. In the one patient here who received placebo, you can see that the signal, the DAT PET signal reduced even further in this individual. Whereas in the lower slides, you can see an example of a patient who received mid-dose CDNF and actually had an increased DAT PET signal after 6 months, indicating a biological response to CDNF. Next slide, please. Okay, so then I thought there would be one slide in between here, but this is the next slide. Okay, so then in the main study, we did find that the DAT PET signal in the mid dose on the group basis, as you can see here in the first part of the main study part of this graph, there in the mid dose group, there was actually an increased DAT PET signal as indicated by the example in the previous slide. During the extension study, there was no further increase in the DAT PET signal in this group. There was actually a reduction by 3.2%. Again, we showed an example of a placebo patient on the previous slide, and here in gray you can see that there is a reduction of the DAT PET signal in the placebo, both in the main study and as expected, as you can see also here with the expected annual decline. For the high dose study, somewhat puzzling in a way, there was a decrease of the DAT PET signal in the main study during the first 6 months. But then there was an increase when the patients were randomized into the main to the extension study. There is actually an increase in the DAT PET signal by 7.4% in the high dose group. And if we compare the original mid-dose group at 12 months with the baseline, we can see that they are 50% above the baseline level, while the high dose is 15% below. Next slide, please. Another exploratory endpoint was the measurements of alpha-synuclein in CSF at baseline after 6 months and 12 months. When it comes to total alpha-synuclein, there was no difference between the groups. So the levels there were no significant differences between placebo or the 2 different doses of CDNF. But when it comes to the oligomeric form, the pathological form of alpha-synuclein in CSF, there is actually a reduction of this oligomeric form of alpha-synuclein CSF by treatment with a high dose of CDNF. And this is interesting in light of the fact that if you compare normal individuals and patients with Parkinson's disease, this oligomeric form of alpha-synuclein has been reported to be increased. So we think that this reduction by CDNF may reflect a biological response. Next slide, please. So here are the conclusions. So this study was designed to show safety and tolerability, and we can conclude that the primary endpoint is met. The study was not designed to show efficacy because there are too small number of patients in the study, and also from what we heard now from Professor Sarema, it's difficult to find a neuroprotective effect of neurotrophic factors in such an advanced population of Parkinson patients as we studied here. However, there were some interesting signals of efficacy. Several patients show a significant improvement in the motor rating of UPDRS, and at least 2 patients showed significant increase of DAT-PET imaging, indicating a biological response of CDNF. And there are also some other interesting cases on different clinical outcome measures, and during these presentations I've shown data from the accelerometer where the bradykinesia score seems to not develop as worse as it does in the vehicle in the mid-dose CDNF, and from the CSF studies of oligomeric alpha-synuclein, the pathological form, where the high dose of CDNF seems to reduce the oligomeric alpha-synuclein. It's also important to mention that the efficacy endpoints also have a safety role, and we didn't see that the patient's Parkinson got worse during the study. So thank you very much for your attention.
Thank you, Magnus and Per. That was very clear. We have a couple of questions. First one is, were there any differences in the results of the study according to gender differences in the results? Let's put it that way.
I don't think that I have access to that data. Perhaps, I mean, Sigrid Booms or someone can comment upon that.
Yeah, but maybe we can just say that there were no clear patterns, and it's also such a small group of individuals. So I don't think we can say that we saw any clear patterns.
Yes, and also I think there were, I mean, it's a very small group of patients, and we had a little bit more males than females in the study. Then we had one question that actually relates to the more preclinical side. That was whether there has been any studies comparing CDNF therapy to stem cell therapies, and as far as I know, no such studies have been published so far. And then we have one more question. This relates to the alpha-synuclein data. Were there any differences in the amount of oligomerized alpha-synuclein at baseline between different groups? It looks like in the placebo group it is much lower compared to CDNF high dose. Well, in general, there's a lot of variability in these type of data, but that should be understood. I can't personally remember that there were very significant differences. I would need to actually look at the raw data to answer that. I don't think we can give you a conclusive answer at this point. Maybe after the break we can come back to this.
I think, I mean, it looks like at least all the data is in the range where you could see differences, reductions in all groups. But it's true that if you look at the graph that I presented, that the high dose CDNF has a higher baseline value, but it looks, I think it's all the groups are within the range where we can see changes in both directions. That's my interpretation of those data.
And also that graph is not absolute levels of oligomeric. It's actually the ratio of oligomer versus total alpha-synuclein. Good. Let's see if we have any other questions. We have a couple of minutes before the break. So there is a good opportunity for the audience to ask questions related to the clinical data. So we have a new question. I will have it. We have a very complicated technical system here. Let me get the question. First question is, has the effect of CDNF been studied in patients with multisystem brain atrophy?
With MSA, yeah.
Yeah, MSA.
Yeah, no, to my knowledge, I mean, not at least in patients. And also that was one exclusion criteria for this trial, to not include such patients. And I think in terms of animal models, I mean, it's there are probably, because the idea with the MSA is there is a lot of alpha-synuclein then in oligodendrocytes, so it's more of an glial pathology, but there are there are fewer studies also on the preclinical level using these models, and they are less, I would say, established than the models that we use to recapitulate Parkinson's disease with. Yeah, but it's very interesting, very valid point, I think. Yeah.
Absolutely, and certainly it hasn't been at least tested in patients, because as the title says, this is the first in man trial with CDNF. But it would be interesting to look at some of the animal models that are more related to MSA, and a very good question in that sense. Then there's another question: is the next step to examine efficacy in less advanced patients? And I think we can, any of us can answer that question. Per, maybe you could start.
No, I think it is, I mean, absolutely necessary before we can conclude, I mean, the neuroprotective potential of CDNF. That needs to be done, and then we expect to see a stronger effect in less advanced patients.
And also I can refer to Heranti's press release a few weeks ago, where we announced the R&D update, where the strategy for the CDNF program is to go for earlier stage patients. So definitely that is a logical next step. And then we have one more question. Thanks to the audience for being active. This is really important. Which one? Yes, there's an interesting question. How long the CDNF positive effects last in people with Parkinson's after the intervention? Of course, maybe I'll start. I mean, we based on the data available from this study, we cannot answer this question. Based on the data available from preclinical models, we know that the effect lasts for about 4 weeks or so. This may also vary a little bit depending on the endpoint that's being assessed.
But we may add also that there is an extension study where we continue to observe the patients now. So we will actually have more information on this point relatively soon. Also in
very good point. And also knowing, you know, the knowledge from the earlier the embryonic graft studies from Lund, based on the long-term follow-up, it looks like actually these type of restorative effects can take years to really show positive effects. So it is very, very important to include the long-term follow-up protocols in these type of studies. Looks like we have more questions. The GDNS, actually, we may come back to the GDNF question later on in the panel. We have Alan also participating. Then there's a question: why do you think the beneficial effect of the high dose seemed to be lower compared to the middle dose? Was it the same in the primate data? Maybe I'll take the last part. Yes, we did see a similar effect in the primate data, or this type of effect, that in a way an inverted U-shaped bell-shaped dose response, that the higher dose actually didn't reach the same level of efficacy as the mid dose or the lower dose. And maybe Per, you can comment a little bit on some of the endpoints that we looked at here.
Yes, I mean, I think for the UPDRS, you are absolutely right that it's the mid dose, and that looks more beneficial also when it comes to the DAT PET. At the same time, we have to remember there are very few patients here and quite a lot of variation among the individuals. And also when we re-randomized the patients, we saw actually that the high dose, for example, then had a better effect on the DAT PET. And also when it came to alpha-synuclein CSF, it's actually the high dose that has the strongest effects. I think it's a little bit premature to draw too strong conclusions. At the same time, I think from a clinical perspective, it's of course attractive if we can have lower doses than higher doses. I mean, to alter, so to say, these biological responses, it's nice to work with a quite efficacious compound.
Then maybe also related to the first part of the question, why do you think there is such an impact? This is not unusual, actually, particularly when we're delivering the compound directly to the target tissue. These type of bell-shaped curves have been observed also for other growth factors with similar settings, and this may also have something to do with the mechanism of action of the compound. So I think people often have an assumption that there's a linear dose response relationship, but that's quite often not the case. So it's not that surprising. Then there's a question: did you measure neurofilament or other markers for neurodegeneration? Related to that, we actually have a CSF proteomics study ongoing. We haven't got the results yet. We expect to get these before the end of the year, hopefully without any further delays with the COVID situation. This is a hypothesis-free screen of proteomic markers that could be differential between the treatment groups, and we will come back to these data when the data becomes available. But specifically, we didn't look at specific markers like NFL in this study. Then I think we're actually quite close to break time. There's one more question. This is actually not a question or more a comment for the whole team. As a person with Parkinson's, with no medical research expertise, I found the graphs confusing. For example, did a higher end value mean that the results were good or adverse? This seemed to vary between slides. Could more easy to understand results graphs be developed? That's certainly a very, very good point. I completely follow the point here, and we do our best next time to have more easy to follow data. Sometimes it's true that some of the endpoint data may be better when the values are increased, and for some endpoint data the improvement may be the opposite. So when values decrease, we'll try to do our best to make easier to read graphs. Thank you very much. So I think we now conclude this session, and it's time for another break.